Hello there. We're already back from the break. Let me introduce myself briefly. I'm Eva Tosco. I'm part of the organizing committee and a member of the Canary Islands Node. I will be moderating this session. Without further ado, it's time to start with the lightning talks. Since the link is already posted on the chat, we have 10 lightning talks about the posters in this session. Each one is 1 minute long, and we don't have enough time for live questions. We wanted to give the presenters a space to show their posters. So if you have questions about any of these posters, please don't hesitate to contact the author. It's always appreciated. You can send them a private message, or you can talk to them during the breaks, since we have the opportunity to contact them at the meeting tables. So they'll probably be there to answer all your questions. So let's get started. If our host can play the video.
I am a PhD student from Spain, and I would like to tell you about my work. The main take-home messages that I would like you to keep in mind are: 1. What is sepsis? Sepsis is a severe systemic inflammatory response to infections. 2. Why is it so important to study this disease? Sepsis has a high mortality rate in adults intensive care units. It's a complex disease with few therapeutic options, few genetic studies, and with no genetics. And 3. How do we study sepsis? We perform the first genome-wide association study of 28 days survival in sepsis that allow us to identify novel targets for sepsis treatment and patient risk stratifications. So please don't miss my poster presentation. I will be very happy to answer all your questions.
Hello, everyone. I'm Eva Tosco Herrera, and thanks to the organizing committee, I'm here to tell you that whole exome sequencing has a great potential in accelerating genetic diagnosis and reducing its economic cost. But a standardized protocol is lacking to manage the great amount of data that it generates. This affects the reliability of the results. So we selected a subset of free and open source applications to benchmark using whole exome sequencing data from 61 patients with known genetic diagnosis. The results show that Exomizer and Amelie have proven a significantly higher success rate as seen in Figure 1 and Table 1. Figure 2 shows how variants can be seen in intermediate files as they were examined for confirmatory purposes, as well as Figure 3 shows coverage and reference sequence limitations of whole exome sequencing. I'll be happy to answer any questions you might have, and I will leave my contact info in the chat. Thank you.
Hello everyone, my name is Eva and I am a PhD student from Spain, and I would like to talk about the work a Fine Mapping Association study of the HLA in sepsis susceptibility. Sepsis is a severe systemic inflammatory response to infection with a high death rate. However, there are few genetic studies and therapeutic options. For this reason, we analyzed the implication of copy number variation in sepsis susceptibility using a genome-wide association study. The results show a deletion in chromosome 6 that is significantly associated with sepsis. This locus harbors the HLA genes. Therefore, here we performed a fine mapping of HLA region given the importance in inflammatory and immunological diseases. So please, for more information, don't hesitate to consult my poster.
Hello, my name is Almudena Neva, and I am going to highlight the most important point of this poster. Firstly, Alzheimer's disease, which is the most common neurodegenerative disease without precise diagnostic method and efficient treatment. Secondly, since sex differences in Alzheimer's disease have been reported, the inclusion of sex perspective could help to a better and faster understanding of this disease. Thirdly, single nucleus approach, which has a great potential to reveal cellular heterogeneity in brain tissue. For these reasons, the main objective of this work is unveiling sex differences in Alzheimer's disease through an integrative single nucleus transcriptomic in silico approach. This study has allowed the characterization of sex-specific and non-sex-specific transcriptomic differences in Alzheimer's disease. These support the need to include the sex perspective in biomedical research, being a fundamental step toward personalized medicine. Thank you for your attention.
Hi, I'm Irene Soler, and I'm going to present the poster A Single-Cell and Single-Nuclei Transcriptomics Approach Reveals Novel Sex Differences in Multiple Sclerosis. Multiple sclerosis is a chronic neurodegenerative and autoimmune condition where sex differences have been reported. However, the molecular mechanisms involved in these dimorphisms are poorly described. In order to unveil them, we have evaluated RNA-seq single-cell and single-nuclei data from human blood and nervous tissues, which were found conducting a systematic review. Afterwards, the selected datasets were individually analyzed, and the results were finally integrated and biologically interpreted. As a result, we have revealed significant sex differences in abundance, transcriptomic profiles, and biological functions in several cell types that could provide a better understanding of the disease and may act as a potential biomarkers to boost personalized medicine. Thank you for listening.
Hello everyone, I am Marina Murillo and I'm going to talk about tRNA Studio. So tRNAs are non-coding RNAs that bring amino acids to the ribosomes in order to perform protein synthesis. tRNAs can be processed into tRNA-derived fragments that perform a wide range of roles beyond protein synthesis. As well, alterations in tRNAs are associated with complex diseases. In order to study tRNA biology, we can use high-throughput sequencing, but tRNA modifications and the sequence similarity between different tRNAs impair the interpretation and the stream analysis of tRNA-seq data. Because of that, we have created tRNA Studio that is implemented into a graphical user interface. It includes a mapping strategy useful to classify and characterize tRNA sequencing data. And as an output, it includes an interactive HTML summary report and an extensive graphical representation of the data, with information for differential expression analysis, plots for tRNA gene coverage that are useful to detect TRFs, and as well plots for the identification and quantification of tRNA modifications. We believe that this work will be useful in order to bring bioinformatics closer to the experimental labs, and as well to expand the knowledge in tRNA biology.
Have you ever used Facebook? Do you know that they use deep learning for suggesting which of your friends should be tagged in a newly uploaded picture? What if we could use deep learning to predict gene interactions from single-cell RNA-seq data? I'm Ina Rivero, a PhD student at TENIC, and under the supervision of Fatima Sanchez-Cabo and Miguel Torres, I'm working on gene regulatory network inference. Gene regulatory networks govern all biological processes, and their understanding is key to fight disease. Single-cell RNA sequencing has shed enormous light on different biological issues and provides a very large amount of data, perfect for the use of deep learning and, more specifically, computer vision. Single-cell RNA-seq, however, is sometimes expensive and not always applicable. Therefore, we examined how generalizable is a convolutional neural network-based algorithm to predict gene regulatory interactions by training and testing it on different cell types. Do you want to know more about what we found out? Head to our poster and take a look.
Hi everybody, I'm Cristina Zamora from the University of Valladolid. I would like to present the first genome-wide identification and characterization of long non-coding RNAs in conifers. Particularly, we studied the Pinus radiata long non-coding RNAs in response to the causal agent of the disease pine pitch canker, Fusarium circinatum. For that, libraries from inoculated and controlled pine seedlings were sequenced, and the bioinformatics workflow represented in the poster was followed. We could identify a total of 13,312 long non-coding RNAs in the pine, being 164 differentially expressed, so consider Fusarium circinatum responsive long non-coding RNAs. We could also predict the potential function of them by analyzing their cis and trans-regulated target genes. So I invite you to see my poster. Thank you.
Hello. I'm Siddhan Sharma, an undergraduate from India, and through this medium of RSG-8 Student Symposium in Bioinformatics, I'll be sharing you the teamwork that we did as a part of virtual research internship experience with HackBio that dealt with enumerating and elucidating the prime-like domains in very interesting families in human proteome. We collected human proteome datasets. We analyzed those datasets through algorithms that are specifically designed to to detect the prime-like domains in proteins, software such as Plaques, software such as PRAMs. So if you want to know about how people can enumerate and elucidate this protein misfolding spaces that forms the prions through computational tools, computational approaches, and building visualizations, do check our poster. Thank you.
Independent component analysis, or ICA, is a matrix decomposition method to solve blind source separation problems. For example, when two people record a podcast, their microphones will record a mixture of their voices weighted by their distances. With ICA, we can separate their voices in an unsupervised manner. Actually, ICA also works great for finding biological modules compared to widely popular methods. To overcome the inherent randomness of this algorithm, we can cluster many iterations of ICA together to obtain robust components. However, this step relies on the choice of clustering method and on computing a potentially large and biased Pearson distance matrix. To address this problem systematically, we created Robustica, a new Python package to compute robust independent components with a fully customizable clustering algorithm and distance metric. With Robustica, we managed to increase the precision, robustness, and memory efficiency of the algorithm. Join my poster to learn how we did it and how you can implement Robustica to dissect your own data.
Okay, so that's it for now. Thank you to all presenters and for all the abstract submissions too. Please remember to get in touch with the authors if you have any other questions. We're going to continue with the rest of the symposium, so we'll close this session and open the next one. So stay tuned and see you all in a minute. Thank you.